Okay, well, welcome everybody. We shall start the session for this afternoon. Please have a seat. Thank you. Okay, so I know some of you like to visit the labs still, the new lab that we just built, and during the break of this lesson, you may like to reach Antonio Cannavacciolo. You know, the guy is sitting next door in front of this room. He's waiting for you in case you want to visit the lab. For this afternoon, I'm glad to introduce, please. Thank you. I'm glad to introduce you the speaker, Alessia Cariani. Alessia, professor of zoology from University of Bologna, campus Ravenna. She's working within the GenoDream Lab, which is a laboratory of genetic and genomics of marine resources. She's running several projects, European projects. I want to mention just one on fishery sustainability of fishery, which is prize fish, which is an interact, I guess. Yes, just finished among the other that you were and are running. So I thank you, Alessia, which is of course a member of our academic board, to be here today for this section. Thank you.
Thank you, Stefano, for inviting me. And
I'm gonna sit here for a while. I'll give the first talk.
Okay, so thank you, Stefano, for giving me this opportunity to join the board of the FishMed PhD. I could follow some of your presentation in the past meeting. I was obviously always online, so thank you for being present here. And thank you for being present on a Friday afternoon, which we are fully aware is not an easy task. So I'm happy to think that Stefano dedicated the Friday afternoon to me, because you know that when the game becomes a bit difficult, the good player.
That's the reason. It's the challenge.
Okay, so yeah, I see many faces that I know, some quite well, some were my past student in Ravenna, so I'm teaching at the master degree in marine biology in Ravenna. I've been teaching there since 10 years, other I don't know. So first of all, I will briefly, 50% di probabilità, I will briefly introduce myself. So I have a very not mainstream background. So I'm an environmental scientist. I'm not a biologist. And I'm quite proud to be an environmental scientist, because now that I think always hear about multidisciplinarity, integration, this is what is environmental science. Now we need to bring all expertise together. And then in my postdoc, I did many things, from microbes to other topics, to cultural heritage. So I've been working on several topics inside and outside academia. And then I was very lucky to re-enter University of Bologna in 2013. And since 2013, I'm in Ravenna at the GenoDream Lab. And these are my main focus. Why I'm telling you this? Because my focus will be then translated in my lecture. So I work on applied science, and applied science cannot exist without basic science. So everything is connected and is very much needed. I like to work in project. I think is a very interesting opportunity. I think many of you have this opportunity, so that their PhD are without big collaborating project, and these are a few that Stefano also mentioned. This is the GenoDream Lab. The lab is not nice, is not in a really nice place in Ravenna, but what is inside the lab is extremely nice. So the colleague and the team and the science that we can make out of the collaboration. So this is the course outline. No? This is the course outline from the statement of the PhD. So really, on a Friday afternoon. So don't worry, I will try to transmit what I know. Maybe it's not even half of what you know. Some are have a very broad knowledge in genomics, some that for example, I'm looking right now, but we will do it together and stop me every time. I will ask you many inputs, and I hope that you will enjoy the end of this very important week that you just had. Starting from you, I introduced myself. I'm not asking you to introduce each of you. You have done it many times in all the meeting, but just a few of you that can think of what can genetics do for fishery and aquaculture. Free flow mind.
It is very useful for stock assessment and discrimination.
Stock discrimination. Can you please tell me what do you mean for stock assessment? Because I'm not an expert of stock assessment.
It means the populations of fish that you are going to exploit via the fisheries, where are they located, and are they connected or not? So
may I rephrase it in stock boundary to delineate the stock boundary and their connectivity?
Yes.
Exactly. This is one of the main points that we will discuss today. Other?
We understand if there are some genetic flows among the population or among the culture that can, I don't know, unificate the DNA and maybe can be a good field for infection and disease inside the aquaculture.
Okay, so this is an application aquaculture related. So you want to monitor the reared fish or reared organism, not obviously fish, and also assess if they have pathogens or other zoonotic causing problem. Okay. I was supposed to type it inside, but I will type it in the bracket when you visit the lab. Other idea?
From changes, yes. For example, the fishery induced changes in the fishery trades. Environmental also not only climate change, but
okay, so fishery-induced evolution, or let's say anthropogenic-induced evolution. We will also discuss this point. So I'm seeing if what I prepare is suitable for you. So if I'm going to reply to your needs or your curiosity. Okay, who does not have an idea what is genetic? Because maybe I started with the wrong assumption. DNA, we all know DNA. Who doesn't have a biological or natural science background here? But you know what is DNA from high school. Okay. And who has quite a good knowledge about genetics and genomics? Who is managing genomic data? Okay, I hope I will not be too boring for you, okay, because we have to be in the middle, no? So from you, any other idea then, now that I spot you, before?
Maybe understand if stocks have the capability to react or adapt to external threat or changes, stuff like that.
Okay, so
the genetic diversity is enough, or
monitoring of the genetic diversity through time. We do it. Okay, so if you have any question or curiosity, if you don't understand, if I speak too slow, too fast, just stop me, okay? I know that you are tired. So, this is what ICES think about what genetic can do for fishery and aquaculture. Everyone knows what ICES is? International Council for exploitation or exploration of the sea. Should be exploration, but I think everyone thinks that is exploitation. But in ICES they have a lot of working group, and there is a working group on application of genetics in fishery and aquaculture. This is the acronym GAFCA. Okay, and this is an extract from their manifesto. So this is exactly the point of view of one of the most important working group. So ICES is managing Atlantic, GFCM is managing, let's say, should be managing the Mediterranean, but there are working group in GFCM too. I don't know if there is a working group in genetics. I've never heard. No. So we look at ICES. Okay, so this is what ICES is putting as main point. No, so you got it right. You can go and work with them because you spoke about connectivity, stock identification, species identification. Don't forget, we will think about this, environmental change, resilience, but also within aquaculture, disease detection, okay, parentage, and in common traceability and interaction within reared and harvested, so wild and animals from aquaculture. I apologize, I have a very animal focus, so I'm a zoologist, so I work with the zoologists. So most of my examples are from, I think all of my examples are from animal. Is anyone working with the algae, seagrass? All with animal? Microbe? Okay. So if you want, since I will leave you this slide, I've put also some link. This is again from the manifesto of ICES, is the new frontier, which are environmental DNA, transcriptomic, adaptive diversity, population size, both from an evolutionary point of view, but also from a census point of view, and epigenetic and transcriptomic as well. Also, journal coming out more and more. This was one of the first, but marine genomics is really exploding in the last 10 years, I would say. And I reported a bit because I'm old and I always look at the past. Yes, I'm old now. I'm definitely old. This is one of the founders in Europe of population genetic applied to fishery, Gary Carvalho. Ever heard of him? So someone said yes, I don't know who. But you maybe you heard it from the lecture, or you have, okay. So he's one of really the founder, and this was an interview. So I'm not going to read it. I just leave it for you. And it's not only Europe, or not only the US. This view is good because it's shared among all the participants. So the Mediterranean, so we are in the FishMed PhD, so let's have a focus on the Mediterranean. The Mediterranean is not only Europe. The Mediterranean is also Asia and Africa. I'm very glad because I had the opportunity in the past to interact with the researcher from North Africa, from Turkey, and very limited thanks to FAO, also to the Middle East country. But this is a poster that is in front of a laboratory in Agadir. So in Morocco, there is a dedicated research center to national research center to marine resources in Rash, Institut National Halieutique. And so you see it is in French because there is their main language beside Arab. There is a version in Arab. I did not I cannot show it. So I took the one in French, and you see that the same topic, the same point of view that is shared also from ICES. So it's good because we have a common baseline, and we can do something which is effective only if we work at the Mediterranean level in a shared perspective. So sometimes we speak about genetics, sometimes we speak about genomics. What's the difference? In theory, no different. It's just the order of magnitude of the number of markers of the power of your analysis. So now the rule of thumb is that genetics is when you have tens or hundreds of markers. And then genomics is when you go above thousands of markers. When I say marker, is mostly single nucleotide polymorphism, so variant. We read the DNA to search for something which is different between this mollusk and this other. Because if we read the DNA and the DNA is the same, we can read the entire genome, but we have no information. No? So we speak about the polymorphism. We read the polymorphism, and that's all. That's the order of magnitude. And why now we can speak about genomics? Because since the early 2000s, there have been a technological revolution that enabled medium lab, people working with medium funding, so no human, no model organism, no clinic, so natural science. They've been enabled to produce thousands of markers. So science is always coupled with technology. No? So advancement in technology enable also advancement in research, in different research fields. And it's a bit of bouncing back and forth because we move from genetics to genomics. No? We want to analyze what we can do with 10 microsatellites. We want to do it with low coverage sequencing for all the individual. But then from a very practical point of view, we need tools. And most likely, then we select the best performing marker to develop small scale assay that can be applied routinely to investigate your population. So, okay, so with the genome-wide resolution now, and also there is a, Alberto, which is the law that say that the more the technological increase, the price per unit decrease. Later. So there is, why this is possible? Because it becomes also cost effective, not only possible, but also achievable budget-wise. But then we genome-wide screen, so we screen the whole genome to then cherry pick maybe 100 markers, 200, that then we can apply routinely. We can apply year after year to monitor the resource, to see, for example, if the disease is increasing or a system of early warning. You want to analyze the tissue every week. You need something which is fast and that you can afford to perform every week. So why I'm focusing on the tool? Because another really important point in my view of genetic and genomic in fishery aquaculture is the creation of solution that can be incorporated, but who has the need? Okay, because there is this very nice review that say we have a problem. Okay, we can do all our genomic magic to find a solution, but do the end user understand the solution? Yes. Okay, we've done a perfect job. Otherwise, all our science is not useless, because it's science, but it doesn't provide the right answer to the problem. So we can define stocks, but if our definition of stock boundary is not suitable to be used as an input to the one that are running stock assessment processes, then they don't talk, we don't talk with the right word to each other, okay? So the course was mentioning an introduction to the methods. I've really shrank it. So, and I'm providing you a few links. So if you want to dig into this, if you want to contact me later, I'm really a nerd, so I really like this topic. But I don't think it's the right topic for you today. So also having the feeling before, I removed a lot of slides from my presentation. So if I'm going too fast, just stop me or, okay. So sequencing, now it's a long history, but it's not that long because it all started Watson and Crick, okay, without, and we should mention Rosalind Franklin, okay. And so Watson and Crick, 150 years ago, so it's not so huge, which is the Locta Volterra, which is, let's say, the basis now of population or stock assessment.
Yeah. In that century, I think in the 30s.
Ah, 1930? Ah, so it's younger. Stock assessment is younger than, I thought it was more. And okay, we have a few Nobel Prize. So Frederick Sanger sequencing, the so-called standard sequencing, maybe the one that you have seen also in cartoon, all the colored peak. DNA has no color, or all when we see this TV series, they are always working with liquid which is colored in tube. No, DNA is very boring, it's transparent. So there are no color in our lab. And you see the big revolution, so you are young, but I hope every one of you was born before the 2000. Someone was born in 2001 too? No, too young, because it's the human genome. Okay, that year is the year of the first human genome, the first publication about human genome sequenced. And so imagine in 20 years now, everyone can have his genome sequenced. Have you ever attempted to run this quick tool, 23andMe? You swab yourself, you send, and they sequence your DNA. And they tell you that you have, for sure, some of your ancestor, which was very famous. So, but the big revolution was, the three big revolutions were the sequencing, the amplification of DNA, is in my next slide, and then what is called the next gen sequencing, the massive sequencing, which started in 2005. And then third generation sequencing only 10 years ago, 2011, 2012. So it's quite a short history. So here I put some faces, okay, so this is much longer, Darwin, Mendel, who doesn't remember? It is Mendel, yes. Watson and Crick, Sanger, and have you ever heard of Kerry Mullis, the inventor of the PCR? It was quite controversial in his last years, in all his life. So he was a chemistry in the 70s. So he was synthesizing lysergic molecule testing, and under one of this experiment, he had the idea of combining different tools that were already available into one system, because it's always like that. The pieces are there, and then there is someone that has a brilliant idea to put all the pieces together. So he also wrote this famous book, Dancing Naked in the Minefield. There are a bit of weird story. And then it's also about money. So economists, I'm always thinking about you. So there is a nice background, economic background, because sequencing the human genome was pushed by what? By the power to improve medical research. But medical comes with money, but also has a big potential, has a big impact on the economy. And so, have you ever heard of Renato Dulbecco and Craig Venter? So they were two competing groups trying to sequence the human genome at the same time. Renato Dulbecco, which was originally from Italy. He was an Italian researcher working in America. He was awarded the Nobel Prize for Medicine for his study about cancer. And he was, let's say, leading the consortium, which was public. And then there was Craig Venter, private. He founded himself Celera Genomics. And then there was all the debate. So the genome from the public consortium was published one day before on Nature, and the genome from Craig Venter one day after on Science. Or maybe it was the other way around. I think it's this way. I remember Craig Venter won, but then the publication was the day after, because then Collins had a secret agreement with Nature instead of Science. So it's kind of, let's say, beautiful, not like soap opera. And also then with the next gen revolution, there has been a lot of economics going on, because companies have been acquired one after the other. Now it's like Zuckerberg having WhatsApp, what does it have now? Instagram, or okay. So it's for everyone, this lecture, not only for me. Okay, so going from DNA sequencing, so normal Sanger sequencing, polymerase reaction, you see 2001, first draft of human genome. What the amount of data produced per run of the machine increase. The price for the same unit of data decrease, but the price per run doesn't decrease now. So producing one kilobyte of sequence, it costs less, but you cannot produce just 1 kilobyte. You produce terabyte of data. So for each sequencing run, the cost, we can afford it now. They are around, I don't know. Recently, we have paid 7,000 euro for one Illumina run, plus VAT. So it's something that even normal lab like our lab can afford, but still it's not 10 euro. Okay? And also the ease of analysis decrease. So you can spend years analyzing the data, because the amount of data is big. So we are in the big data period, not only for genetic. And this is really a graph that shows you the trend is really increasing. I cannot say it in English. La pendenza della curva, the slope, okay, not the trend, it really increases exponentially. So in number of data produced per day, and you see in the last 30 years. Again, more detail if you want. The link brings you to very easy web page, not very complicated. So this is what are called next generation. Is not really next, is now for most of them, and for other is no more, because some of them are dead. Other are still zombie. no? Ion proton, maybe some approach. So I will skip all the approaches that are dead or near to be dead, and I will focus only on what is the big difference? Is the amount of data produced, but why we can produce this amount of data? The steps are the same. You take a piece of DNA, eukaryotic, prokaryotic, whatever, a piece of DNA, or the complete genome, but then you fragment, and then you need to amplify. But you don't amplify one fragment at a time. So you still amplify one fragment at a time, but you parallelize it. So in synchrony, you amplify millions of fragments, and you sequence at the same time millions of fragments. That's wonderful, perfect, but there is a but. These sequences are short. Most of them are short. So it's a bit complicated because Sanger sequencing, the normal sequencing, has two advantages: that most of the time you know what you are sequencing. You can do shotgun Sanger, but most likely you know the target of your sequencing. So it's like creating, imagine you have to recreate a big puzzle. And you know exactly which pieces of the puzzle you have. So it's very easy then to put it in the right place, even if you don't have the other pieces around. But you know, this is blue, I have only one blue cloud. The blue cloud is in this corner, I put it here. And you can see that the piece is blue. You can read it. The user can assess the outcome of the sequencing. Here, no, because it's too much, and because the way the data are produced, they are image or they are recording of change in the voltage of ion or in light emission. So there is no data that you can visually inspect. You have to base your analysis on quality statistic. So, and also, you can still know that there is a blue cloud in your puzzle, but the pieces that you have in mind are so small, so you don't know which one is in which corner of the cloud. So reconstructing the puzzle, which is assembling the information, the genome, is very difficult. So this is all the technique that I'm going to say today. So again, I'm focusing now only on what is really used, what you can find in paper, or what you can read. For if in your PhD you need to sequence a microbiome of your organism, or the microbiome of the environment in which your organism is living or you are rearing. In lab, 99, I bet a beer that you will use Illumina sequencing. If in your PhD project you are not going to use Illumina sequence, you call me, Alessia, there is a beer to pay. And because it's the market. They are taking all the market because they were smart, buying other company, outcompete all the other companies. So now Illumina is the main sequence provider for next generation sequencing. And also the price, they are very good in their marketing. So they show this nice graph in which the cost per gigabase decrease, the output increase. Then if you multiply, it rests the same. No. It costs less, but it produces more every time. You can go to the butcher and say, quant'è la fiorentina al chilo? Ok, però me ne taglia 20 grammi. No, sorry I cannot translate. And that's why. So a brief how Illumina work. So you fragment your DNA. Remember, fragment, because then the next generation, we don't fragment anymore. We fragment, and we amplify. So each little piece has to be replicated many times because you need to read it many times. Okay, how it gets amplified on solid state, surface, bridge amplification, it's all Illumina patent. Forget about it. You are not in a PhD of molecular biology. And then it produces sequence data, which is based on color. So it records color. So there are a lot of screenshots, imagine like screenshot, and then from something which is called flogram, it reconstructed the sequence. And then you have to align. So you see, this is the true sequence, but all your fragments are short. They don't start all at the same time and all at the same time. So you have to reconstruct the puzzle and see, for example, if you have variant with respect to a reference or if it's a sequencing error. So it's very complicated, the quality check on this data. The good thing is that even if they read, so when we speak about next generation sequencing, the sequences are called reads, le letture. Okay. Even if they are short, you know they are always paired, and you know more or less the distance between the two. So you have those two tiny pieces of the puzzle. You don't know where they end up, but you know that wherever they end up, they need to be 200 base pair nearby. And this is important to reconstruct the puzzle. So what is really next or new, or at least now, is the third generation sequencing. The approach to sequence in which you don't have any more fragmentation and you don't have any more amplification. So you sequence, in theory, a complete molecule, a complete chromosome. Is still not the case, but still very long sequence. And we have mainly two companies which we could invest on. They will not be by one by the other, I think, now, because there were other companies like Helicos or many other spin-off that then didn't make it. So now we have PacBio, Pacific Bioscience, and ONT, Oxford Nanopore MT Technology. Non mi tornava, I could not get the T, so ONT is Oxford Nanopore Technology. So they both sequence long molecule. So you don't have to fragment your DNA. You need super high quality DNA, so super long molecule, and they don't amplify it before. So they really sequence real time. One is using a single molecule real-time sequencing. So everything is under patent, is their technology. And the other is sequencing by passing the molecule to a modified nanopore, so a modified protein. So they tell a bit what they are doing, so it's not a secret, but they don't tell everything. But the difference is that one machine is huge. PacBio is huge, and it needs also a specific, you cannot put it everywhere because it's very sensitive. You cannot have vibration because the system of detection is very sensitive. And the other machine, which is doing exactly the same thing, is USB size. The very small one, you can have it bigger, but the portable one is really this size. And I can tell you that in Ravenna, one group has it. And this is the size. This is a picture I took in Ravenna. So this is the device. It's really very small. So
how much it costs?
The device, not that much. It's like the printer that you have, at least the printer that I have at home. The printer costs maybe 40 euro, but then the toner, the cartouche costs maybe 100. So at Oxford Nanopore, so there was a period in which they were providing for free the mini ion, this one. This is called SMIDge ion, I think, but then it costs a lot, the kit, because this is what you need for the very last process before you need other machine. But this is what they are selling now, and it's indeed very small. You can still use it if you have like a small lab in the field. They go to expedition in Amazonia and they barcode all the insects that they are discovering. Oh, it's true. We want to put one of these on a submarine drone. It's just a matter that our proposals are rejected, but we are still applying for having one and sequence real time under the water. So and you see, so this is Illumina sequence, and this is the other long sequence. So you say, oh, let's all go for long sequence. Why we still need Illumina? Error rate. These two methods still have a very high error rate. So they provide you a very long sequence that you cannot really trust, but it's very useful because it gives you, it's like that you have your puzzle. I'm sorry for the one that attended my course in Ravenna, because I'm always using this puzzle example, but for me it's very effective. So I'm sorry to Letizia and Federico. You still have your puzzle, you still have your tiny pieces, but with this other machine, you can produce a very maybe blurred, low resolution, big piece of the puzzle that you can use like a scaffold to then fine-tune your sequencing. Okay, so now most of the project use long read. In the meantime, long read accuracy has improved. Now there is a PacBio new kit that say that has a very high accuracy, is what they use to sequence genome. But then there is always some need for fine-tune with other information, or Illumina sequencing, or other optical mapping, other things which are very specific for genomics. So I consider the technical part closed. I know that not even an introduction, was just really a glimpse. But for me, it's more important that you have a take-home message instead of the technicalities. So what next generation means high throughput. High throughput sequencing, so both short read or long read, really enabled many new research that were not even possible, you could not even think about 10 years ago. Okay, so because sequencing is becoming cheaper and cheaper and faster, and we can say that now the sequencing is only limited by our imagination, even if still we have some technical and budget constraint. No? So we all have limited resource. No, ecology tells us that we have limited resource. So we can sequence whole genome de novo. So we sequence an animal, a plant, an organism that was not sequenced before, or we resequence. No, human are, when you say you can sequence a human genome in 1 day for 100 euros, it's not sequencing a human genome, it's just a resequencing, just maybe reading one time each base and then comparing to the human reference genome. We can then do comparative genomic, epigenetics, or we can sequence only the exome or the transcript, so the mRNA or all other kind of RNA molecule. We can sequence pool of genome. If we are not interested in the information at the individual level, but we want to contrast Daphnia exposed to copper and Daphnia individuals not exposed to copper, you sequence maybe pool of 20 individuals, 30 individuals. Or you can even pull, you can even sequence just a teeny slice of hopefully representative of the genome of hundreds of individuals. And then you can do population genomic. You sequence fish from 30 locations all around, from all marine protected area of the Mediterranean. And you see then if there is connectivity. Or you can sequence from water, from soil, from gut content, from, you can take an holobiont, mix it, and then sequence everything which is in there, no? So all that glitters, so it's fantastic. Why we don't do it every day? Because it costs, and also because all that glitters need data analysis. So the amount of data produced is enormous, and it requires, there are still, even if new script, new methods, new computational approach are developed, and it's very nice because it's quite always free, open source code is still challenging the analysis of genomic data. There are some topics, let's say, or some applications which are more streamlined. For example, microbial fingerprinting. There are a few tools, let's say, for dummy. But then if you really want to enter into the core of the research, I think is very difficult still, no? Okay, you are the microbial.
Yes. It's not so easy.
Exactly. Instead, even if it's the first research field in which this all started, because I remember there was a very ARB. Have you ever heard of?
No.
It was the first repository of all 16S bacterial sequence. So it all started from there. The metagenomics started from prokaryotes, but it's still quite challenging. And so, yes, just to let you know, but I imagine that even general linear model is not easy to understand. Stock assessment, no, some tools for stock assessment are very complicated. I cannot imagine about the model and underlying economic forecast. So we need to analyze data. We are scientists, no? We need p-value. We need result that we can trust. We cannot forget about the conceptual framework. No matter the technique, no matter the power, the terabyte of genome that we can produce, if we started with the wrong conceptual framework, the wrong sampling design to test our hypothesis, no way that we can solve it with statistics or with more data. So that's why I propose you also this image from a paper of 2016. So always think about which is your scientific question and which is the sampling design, or even if you don't go out and sample, which is the experimental design that you need. And then you can start, after that, you can start digging into the very technicalities. But if you didn't focus enough on the starting point, like if your houses, the pillar of your home are not solid enough, no matter how fancy the architect will create your home, it will not be robust. Here a few information, a few examples, but also a very interesting winter school that was running in Switzerland for a few years. I don't know if with COVID was stopped, maybe they will restart it, but all the lesson, all the tutorial are, as I said, open. Everything is on GitHub. So this is the link. It was a very nice school on adaptation genomics, comparative genomics. I don't know if you have ever heard of it. And it's nice because all the resources are available. Can we stop? 5 minutes? Also, because if you want to take the tour of the lab, I don't want the colleague to wait too long. Maybe the colleague wants to go home. Sorry, question? Could you...
application for machine learning?
Yes, but it's more for functional annotation than for... you sequence the genome. I don't recall now any artificial intelligence AI. Now you were mentioning for the assembly of the genome, but then you want to find a meaning out of the genome. Now you want to know in this genome which are the coding region, which are the promoter, which are intronic, which is what is called junk DNA, which is not junk, and so I'm aware of artificial intelligence application to identify and provide a putative function to the genome. Well, I don't know, maybe there are other applications for community reconstruction, for example, for bacteria. Now maybe you have tool to reconstruct a bacterial community. Now you have different functional group, and maybe I imagine this can be an application. Okay. During the break, I stop the recording.
No, just a bit.
Okay. Okay, I will restart, but take your time to finish the food, the drink, and feel free to go and take more because you have to finish everything, okay? So, I imagine it's allowed to eat in this room, okay? So you can eat, like in workshop, you can go and take a cup of coffee and come back and listen and interact. So keep the sugar level high. So I think we can consider it achieved, no? The recent advance in genome technology. And the main message is we have a very nice technology, so we can all use this technology, because I forgot to mention that beside the company, there is also a huge market of service provider of this tool. So you can do genomics even without a molecular lab. You can take your sample and ship it and have it DNA extracted, assessed, sequenced. And then they will send you data. But then you still need to analyze the data. That's why the take-home message is like this. Sudo, make me a sandwich. Who knows what is sudo? Super user do, no? So, okay, all of you could get the joke. I'm sorry, but it's a joke for those who are used to script. So there is a lot of scripting. A very few tools have a graphical interface. Most of the genomic data analysis by script in Unix, so simple Bash or Perl or Python or other languages. And there is no way to escape R. I think every one of you will face R, no? The computational, which is very useful. Okay, so let's enter into the topic. FishMed PhD, so I'm sorry, I say this really animal-oriented and harvesting-oriented. So there is this cornerstone in the field is this review from Fish and Fishery, is a very relevant journal in the field. which was produced by researchers from Cairo. So the research center from Australia now is usually they are very good. They're always ahead of time, so they always start a new way of thinking. And this review from Jennifer Ovenden and colleague is really a milestone. That's why I'm going to present it to you. And you say, okay, but it's from 2015. We are in the big era data. Something that was published last year is just outdated. Yes, it's true. But it was from Cairo, which normally they are 10 years ahead of us. And I will present you the recent implementation in the Mediterranean. So, is a review. Okay, so from Queensland School, the University of Queensland and Cairo, and because it's a review, is nearly 30 pages long. Do you think fishery scientists will ever read such paper? Do you think you will ever read such a paper? Unless you are very obliged to. No, so that's why they were brilliant at Cairo. Sorry. They were also very with an attractive title, no? Ocean 11. And at the time, it was just Ocean 11, then Ocean 12, Ocean 13 came later. But the very good one is this one. Ocean 11 is already a reboot. I didn't know, but the first Ocean 11 with Frank Sinatra starring on it. So it was very old. What they did at Cairo, because Cairo is not only academic, so it's really management agency, is also management agency. They translated the review, they published the review, and they translated in a field guide to genetics in fisheries. So very plain language, very direct. What I was mentioning before, we can have the technological solution from genomic, but is the end user really uptaking it? If it's not, we have the solution that doesn't solve the problem. Okay, so they translated in more concise. Is a leaflet, is very. I have the PDF if you want to print it, but you can find the PDF online. And they identified 11 topics. That's why Ocean 11. Divided in main group, so understanding the attribute of the species which are harvested, measuring the impact of fishing, biosecurity tool, and post-harvest. So we'll quickly go through this. We can have a last break at the end of this presentation, and then we can have a quick wrap-up. I was hoping to finish by 6, because I wanted to give you the time to go back home. So if I'm going too fast, just stop me, okay? So species identification, come on. Who cannot recognize a fish? Besides Federico, everyone. Federico Calì. I don't know if Federico Maioli can recognize. I cannot. I only see small amount of tissue, small amount of fin clip. And then we extract DNA, and all the tubes are the same. You have no idea of how many genetic data are produced with the wrong label. So associated to the wrong species. So species identification is really important. Taxonomists are at risk of local extinction. And for a series of reasons, and that's why it's important to have molecular taxonomy. But molecular taxonomy is not enough. We need integrated taxonomy. So molecular data must be produced together with the taxonomies. Okay. Otherwise, we still work in two separate worlds that don't help each other. It's difficult for fish, especially when they are processed. I cannot imagine for invertebrates, no? polychaete, or they're all warm, they look the same. I hope no one which is an expert in polychaete is listening. So why I'm here? Because the team, not me. The team I work has quite a few experience in this. So we started in 2008 with the molecular taxonomy, with an approach which is called also barcoding of life. I hope you have heard of it. Is a worldwide initiative, not American. For once, the core is in Canada at the University of Guelph, and they want to produce molecular data able to identify to species level each specimen. So we don't identify species with molecular. Okay, identify a species is something different. Sorry for the terminology. You assign a specimen to a species. Did you get the slide different? Okay. In italiano, sorry, non scopriamo le specie, non identifichiamo una nuova specie. Per identificare una nuova specie ci vuole l'olotipo, ci vogliono un sacco di cose. Noi assegniamo un individuo a una specie, ok? E lo possiamo fare, why we need to do it? Food control. Once we have fish fillet, it's almost impossible to recognize them. You can have sole, plaice, flounder. They all look the same. Imagine that it's very hard to separate blue shark from swordfish, verdesca e pesce spada. Molto spesso vengono, many times you can have substitution fraud between a teleost and a Erasmus. So fish don't carry the ID, but the DNA can tell you this specimen is ABC, as long as you have standard, so reference sequence for A, B, C, D, and all the species that you might encounter. So this is, for those that haven't seen an electropherogram, a sequence is like this. Just pick, colored pick, that you then translate in a sequence of A, C, T, G, and so on. So there is this initiative, which this is a screenshot of the fish barcode of life, but we have marine barcode of life, but now it's really barcode of I, not from Apple, but is international barcode of life. So it's really worldwide, and they are attempting to produce a reference sequence for all recognized species. Species is something which is a concept which is better suited for animal, even if it's a bit tricky. Also with animal, is more difficult for, is more challenging for plant. When you enter prokaryote, is very difficult. No, you can speak about strain or so, and also in that case they have horizontal gene transfer. So molecular typing, you don't do taxonomy. Now you do molecular typing for prokaryotes. So prokaryote is 1, 2. Who is working? 1, 2. Chi è che lavora con I batteri? Okay, so I'm trying to translate the same concept to match everyone's needs. Okay, this was my first European project, fish and chips. And to cheap, it's a very quick tool to recognize a species without sequencing. So like the COVID test, no? Now they have this flow, I don't know the word, but really the same format of the COVID test that we all did more than once in the past years. They have it, for example, to recognize endangered shark species. There are, for example, some hammer shark which are endangered, other which are less endangered, but so you can fish them. And someone created a very quick assay that you can run on one of these lateral flow tests, and they can tell you, depending on the band base, if it's one species or the other, and you don't need to sequence or anything. It's really real time. And we have this initiative, which is still going on. Now it's called version 2. It's called Elasmodrop because we analyze environmental DNA to try to find, we will analyze environmental DNA for elasmobranch conservation, but we first need to map, to create a standard for all the elasmobranch. And when we started doing this, we discovered a lot of misidentification. But misidentification not carried out by me, I did a lot in my previous life. I did a lot of survey, but I was just moving stuff. So I was aware that I was not good in recognizing species. These identifications were really performed, but very experienced colleague, but simply some fish are really difficult to understand, especially juvenile state, especially elasmobranch, rays or Mustelus punctulatus. As long as they are not maybe 50, 60 centimeter long, punctulatus doesn't have the dot. So imagine the landing. Now, if you work with the landing data, you don't know, it's just Mustelus. You cannot say which Mustelus is. And you see quite a lot. And also, especially for shark, every morning, a researcher wake up and think he discovered a new shark. And so, Squalus megalops, Squalus, and so a plethora of name for the same species. So it's really confusing. But you can, have you ever tried to taxonomically identify early life stages, so larvae? Is it easy?
We have a workshop at the end of the month.
On which kind of larvae?
Ichthyophaena in general.
Okay, so.
Yeah, I was looking for it for several years, and then there is Lorenzo Cernelli from Istruzione Zoologica, coming from the US. After 20 years of research only on this, because in Italy.
No, in Italy, no. And for sgombrid larvae, EAO from the Balearic, they are very good. No. But also my former colleague, Greg Panzer, who did this work, he was really going in search for the taxonomic table, and they were wrong. So the name were wrong on this, so it's really hard. So with DNA, you can identify larvae. And you can identify, we managed to extract DNA from one A ball of the larvae so that we could maintain the rest of the larvae, because if you destroy the larvae, so we could. Or bones remain, fragments. When you don't have any taxonomic key for identify the organism. DNA is still there, fragmented, degraded, and so on, but still you can perform methods for integrated molecular taxonomy. Or even before that early life stages, have you ever seen this? No? They are called mermaid purse, il borsellino delle sirene.
Yeah, a couple of people went to the beach 2 days ago and they found some.
Put your laptop in a more safer place. So this is very important because if you find this, it means it's a nursery area. It means some animal was there and left the eggs. These are eggs of rays or shark. These are from rays. So identify them is very difficult if you don't find the embryo inside, but it's possible to extract DNA from the material inside the capsule and try to identify the species. And we could use this to identify potential nursery area or potential sensitive habitat. Maybe using this terminology is something that you have heard, no? Vulnerable habitat, sensitive habitat, to protect or to study better. So who was speaking about stock definition, stock boundary? This is the topic in which the group I work with has the most long story experience. So, and we are still running project on this because, oh, sorry, I forgot to translate. Because despite what Axley was saying, fish are not the same everywhere. Fish are different. They are less different than terrestrial species. We don't have mountain, we don't have river that cannot be crossed, are less differentiated than freshwater fishes. You can trace each salmon to its stream, no, because salmon go back to freshwater, but still there is population differentiation also in marine fish. And also in marine fish which have a very high census size, so they have less genetic drift than other species. We simply need better tool to investigate. If you want to see something which is very small, you need to turn your microscope or your lamp to a better magnifying power. And so that's why it's more than 15 years that we work on population genetic or genomic to infer population structure of marine animals, mainly fish, but not only fish. So it started with this EU project, which was a very big EU project, in which we were asked to provide maps, provide something which is quick, understandable, that can be really used during to decision, that can be provided and can be easily interpreted by decision maker. So beside this map, there is a lot of genomic. The color are not by chance. The color is the inferent ancestry of the genomic cluster that could that we analyzed in this fish. So we did a lot of next generation sequencing. At the time, it was really the beginning of the technology, so it was throughput, but not so high throughput. Let's say we scanned the genome to pick the markers that were mostly that showed the best discriminatory power. So the problem in marine fish is that there is a lot of gene flow. There are barriers, but are not the barrier that we are used to in that people studying terrestrial animal were facing. So we need to pick only the best marker that tell us more and leave the rest outside. Otherwise, it's like having a fog which is blurring everything. No? So we go and we search for outlier marker, and use this outlier marker, we can really have a better resolution of the differentiation existing between our geographical sample that might reflect difference in different stock, so we can identify stock boundary, and ultimately we can have also different biological population. So stock is what you exploit from the biological population. Is the correct definition? Stock is a management concept, no? Not a biological concept. No.
More the way in which it is used is usually more related to management, but theoretically defines some population that is like some way separated those population.
So the best is that you have one biological population that you manage as one stock. Yes. Okay. So, but sometimes you manage.
Most of the time, it's not really respecting the actual genetic stuff, because you have to manage stuff according to some geopolitical areas.
But so that's why it's important to see the boundary of this stock. So maybe you have to manage two together or split one in two, because it depends. But so this is I'm showing you the result of the most, let's say, important species for Mediterranean fisheries. So is hake, Merluccius merluccius. Who is doing his PhD on hake? No? Stock assessment on hake? No, nessuno. Non è più importante, it's not an And again, hake, so genetic is a tool, is not the only tool, and is not the magic wand of Albus Silente. It cannot solve everything. It's not the elder wand, no, la bacchetta di Sambuco. So we integrate, no, multidisciplinarity, integration. So genetic can tell you something about reproduction, but also it gives you an information in a time frame which is not the time frame of the management, because genetic tells you something of generation. If you want to know the exact spawning biomass or other, you need different source of information which are in a different time frame and in a different also geographical grid. You cannot genotype all the fish that you get from a survey. You can measure all the fish that you get on the survey, length and weight is really time consuming, but sometimes you have to. So this is why genetic is nowadays taken into account and merge with other layer of information to create identification of stock unit and stock boundaries based on multi-criteria approach. So once you have maybe resolved your stock, you manage, you have completely aligned your biological population with your exploited stock. There are some situations, for example, in the middle of the Atlantic or in other particular area in the Mediterranean, in which you have different fishing fleet insisting on different population or different than on different stock. Imagine that you have area in which fish aggregate to feed. And it's very important for quotas and stock assessment that you can somehow resolve what is called mixed stock fishery. This again, it's very important when you have quotas. In the Mediterranean, we still don't have quotas for the majority of the species. As far as I know, we have quotas only for bluefin tuna. Can colleague correct me? Maybe we will have quota for small pelagic soon.
Not yet.
Which other?
I am aware only for large pelagics, but in the last days, some of the teachers mentioned quotas for other species. I was not really aware, but
so other species in the Mediterranean have landing quotas? Gli spada?
It's not a quota that actually now for pelagic.
Can you repeat it loud?
I think small pelagics have actually since
24
years. Yeah.
Okay, small pelagic.
Yeah.
Okay, and yeah. At the moment, the only one which is managed with the quota internationally is bluefin tuna. That's why it's very important mixed stock assessment. So resolve mixed stock fishery. Have you ever? I don't know if you know, but do you know what are regional fishery management organization? No? See, maybe. Okay. So we have supranational organization which manage fishery. In the Mediterranean, we have GFCM. in the Atlantic for tuna-like species, now also shark, we have ICAT. So ICAT is financing since 2010 research on mixed stock assessment of bluefin tuna. It is very important because it moves a huge economy, tuna. Tuna affect economy, tuna affect politics also. Relationship between USA and Japan, Italy, European Union and other countries. So it's not only about science or resource conservation. So we were lucky to be involved in this project for 8 years. We are not involved in it anymore. And we had a huge sampling. It's not trivial to get tuna. You have to pay for the tuna. And it's not trivial to have a small individual. Because if you get a tuna big like this in the Ligurian Sea, can you tell it comes from the Ligurian Sea? No, it can come from everywhere. Bluefin tuna can take transatlantic migration. Tagging experiment have demonstrated they can take transatlantic migration after the first year of life, in which they are 30 centimeters, more or less. So if you want to assess the genetic component, if you want to associate a genetic fingerprinting with a geographical area, you need to be sure that the individual has spent most of the time in that area. And so with tuna, you can only do it if you sample early life stage, small tuna or larvae. Larvae are very difficult to catch and to identify. We were supposed to analyze 75 larvae from Eastern Mediterranean. It's 20 years that the Turkish colleagues say that there is a spawning area there because they get larvae. We analyzed all the larvae and only 3 of them were tuna. All the other was something else. It can be by chance because tuna is very opportunistic. It's a batch spawner, it spawns everywhere. When conditioned, but still 3 were tuna. But, so we say, okay, larvae, maybe no. Can we get the juvenile? At least we are sure about the species. So for juvenile, you need a special permit because it's illegal to fish young tuna. But under ICAT umbrella, we could do it. So we could analyze from larvae to young of the year, medium and large adult. And this is the difference. I'm entering into technicalities. I know, sorry. Don't worry. This is the bar plot. The color reflects how much of a genetic component you are made of. So what you can understand from here, you have all Mediterranean individuals, which are not totally, but mainly associated to the light blue genetic component and the Gulf of Mexico, so the other side of the Atlantic, the other component. Sorry, I forgot to tell that tuna is managed as 2 stocks, and the 2 stocks are identified by throwing a line in the middle of the Atlantic. So what is this side is 1 stock, what is the other side is another stock. Tuna can move. No? But they say that there are 2 main reproductive areas, one in the Gulf of Mexico, one in the Mediterranean. Nowadays they discover at least other 3 reproductive areas. Slope Sea, Azores, and they say also Bay of Biscay. So with the genetic, you can separate if you find difference, no? So here are the difference. They are very, very low, but significant. And so if you have something which is different, you can then assign. You are more light blue or dark blue, depending on your genetic composition. And this is what you do in mixed stock assignment. You assign fish to one or the other, or 3 or more, depending on the situation. And this is the assignment assignment result that we obtain. So these are the young of the year, so the early life stages, and you see that the Western Atlantic, they do have a majority of Western Atlantic origin. So indeed, they are quite different from the small fish, small tuna from the Mediterranean. If you want to, so these are the larvae or the young of the year. If you then want to assign the feeding aggregates, so the places where you find adult tuna which are predating, which are feeding, then you can see that the components are different. There are some areas in which you have more Western Atlantic origin and other that you have more Mediterranean origin. So if a fleet has quota for Mediterranean stock, depending where it goes fishing, maybe his quota is insisting on the Western, let's say European and American, because these are the two big players. Japan is buying quota from both, so they have all quota. So genetic is a genetic fingerprinting trying to assign something to a putative stock of origin. So you see, it's not that all clear cut. Many individuals are misassigned because most likely we don't have true biological population. We have a system of meta-population. We have more population that we didn't sample because tuna is reproducing also. Otello. Good afternoon. That's it. Tuna is reproduction also in other area. So we are not working for ICAT anymore, maybe hopefully in the future. Other colleagues are still working. ICAT will continue, the management of tuna will continue, and ICAT will still continue financing research, not because they are very good, because they must do, because there is a lot of fuss about tuna. And a paper published by other colleagues exactly the year after our paper found exactly the same result, you see, including the slope C reproductive area. But there is a mixed stock analysis for cod, because for cod, you know that there are different ecotypes, different populations. So this is something which is very useful in the management perspective. Who is doing stock assessment beside Enrico? Is someone else doing stock assessment? So you need the strata, no? Age 1 with length. Is it easy to identify? Do you have a very robust age at length key?
Absolutely not. For starting, especially, it's a mess, totally mess. We have a meeting 2 weeks ago that they redo the reading of the algorithm, because the operation LK are different from Italians, are different from Slovenians, are different from so they have this rereading and probably they agree kinda, but the growing parameter are not good still, so yes.
I know it's very difficult. I'm not doing it, but everyone tell on which species you do the.
From crustaceans, so I do not have automated age by length.
By length. So it's the population structure. So the age classes is a crucial parameter now in stock assessment, but it's very difficult to estimate. So someone say, genetic. Can you please help me with your magic touch? Is not, but is promising, let's say. And so again, I have the experience of tuna, and you all know Marco Stagioni. So Marco Stagioni is a brilliant scientist, and he was the only from Unibo and another colleague which is not here anymore, not working in Unibo anymore. They were all having a meeting for reading. In this case is otolith, but it's also the spine, I think, yes. Otolith are not used anymore for tuna, now they use the first spine, but still it's very hard to find an agreement to define this ALC, age. So, Professor Luiselli is gone, but he is on her, I took her work. So again, natural science, we stem from human. So we want to know everything for human because we want to live together, live forever, not together maybe sometimes, live forever, live healthy. So everything we know from genetic is from the clinical point of view, no? So, but then we can take the best and apply to natural science. So we have a study on methylation pattern. So epigenetics, the modification which are supposed to be not inherited, but maybe they are. So epigenetic is a very intriguing field. If I can turn back time, I would like to do a PhD on epigenetic. And so they estimate some markers, so some region of the genome in which the pattern of methylation is correlated with chronological age in human. So they use it, for example, also in forensic to understand the period of death. Yeah, sometimes a bit creepy. So, but these are work from a colleague from the University of Bologna. And then from human, you can translate it to marine animal, mammals, no? The closest. So again, from Cairo, they started this group, which is called Enviromics. And they want to do, they are doing study to estimate epigenetic age. For example, they started with mammals, so cetaceans. And now they also try to do it with whale shark. It's also quite easy because you can easily collect biopsy from the animal 1 year after the other, no? Because you can mark, tag, and recapture whale shark. Now they are all monitored, like with photo identification, and are also easy to approach. Moving to wild, to fishes, is more challenging, but it's possible. With the genome-wide approach, it was possible. There is a very, so the expert in this is this group by Francesc Pi Ferrer, I think he's Catalan, which managed to do it on cod, I think. See, or cod or egg, because it was possible for him to rear animal and to sample animal at different time point. Then you can argue that maybe the methylation pattern in captivity is different from wild, but still is a first approach, no? Because otherwise there's no way not to, beside otolith, and colleagues are telling me that, for example, in the Baltic, because of all the environmental condition, the otolith ring are blurred. No, ma è quel punto lì. I computer sono in salvo. So there is potential. Genetic can provide useful tools or can work in this direction. And then we have ecosystem monitoring, no? Assess the biodiversity, the composition, the so-called sorting. I did it in my, and I still remember it, sorting of animal. So really, we really already have this tool. This tool can be easily implemented. The point is how accurate are the tools. It depends on how accurate is our reference database. It's like chromatography or mass spectrometry. You identify a molecule only if you have the standard of this molecule. Otherwise, you see a peak and you cannot put a label to this peak. So, you can use this approach to, for example, analyze traffic web, gut content. I did gut content analysis morphological, it's impossible. So with DNA, you can have a tool. It's not 100% sure that is better, because DNA also degrades in the stomach. So you will never be sure that you are taking 100%, that your accuracy is 100%, especially in wild animal. So there were studies of proof of concept in animal in captivity, like seals, which were giving, the researcher knew what the seal was eating because they were feeding the seal, and then they were analyzing not the gut content, the fecal content, because analyzing the gut content is usually lethal for the organism. And so there is this study about ache, again, in the Adriatic Sea, with a metabarcoding approach. So you take the gut content, so everything which is in the stomach, so all the prey undigested, you extract everything, all the DNA, you treat it in a way that you block the host, otherwise you sequence just the ache and not the, even if in this case a problem, because if there is cannibalism, then it's a bit complicated, but still you can do it, and you can analyze which animal, which organism you find in the gut, in the stomach content, and you can then do it at different level of the traffic web and reconstruct the food web. But what is metabarcoding nowadays used every second? So there is a paper on environmental DNA really every second now, especially in fishery science, is because environmental DNA is very useful for biodiversity assessment from air. They are doing pollen metabarcoding from air, soil, and from water. It was environmental DNA was first developed for freshwater monitoring. So searching for specific amphibians in freshwater. But you can do it also in salty water. It's more difficult, but we can do it. And it's very important to couple it for ecosystem monitoring and fishery management. So these are the very first two papers about environmental DNA in marine water, but now there are really, I cannot tell you which is the most outstanding. There is a review on environmental DNA really every day, a special issue. Now there is even a journal which is called Environmental DNA. And as a review, I would suggest this one, which is called the Skeptical Optimist. So it's kind of analyzing pro and con. And there are very specialized groups in environmental DNA in Italy. For marine, not that much. The Stazione Zoologica is starting in the Palermo Center. Giorgio Aglieri, Carlo Cattaneo, everyone is working with them. No, anyone is working with them, okay? Otherwise, there is Italian scientist working abroad, Stefano Mariani, which is one of the top scientists in this field. We are trying to do with our best researcher.
Best of all the time.
Yes, the goat, no? So this is I put again, it's Friday afternoon for everyone, also for me. Your week has been very tough, mine not, but not even easy. So we are laughing because the researcher in the picture, so the one that is not a fish, the animal that is not a fish, is a colleague of some of us, because he was studying in Ravenna with some of your other PhD colleague. So, okay, environmental DNA, water. You need to take water, many liters, and filter. And filter as soon as you collect it, because DNA in the water is degraded and is degrading because there are bacterial, viruses. DNA is not meant to stay in water. DNA is meant to be degraded, no? The cycle of nutrients and of elements. So is not persistent. So you have to filter it quite fast. You can do a survey, or you can ask, there are researchers from Milano Bicocca which have an agreement with ferries. They filter water in the ferries, so they continually monitor Livorno, Olbia, Livorno, Punta Aranci, so the ferry path to monitor, for example, for pelagic, for cetacean, because il santuario dei grandi pelagici. Or colleague, not us, smarter colleague than us, developed this way to sample water and the DNA inside the water in a way which is highly compatible with fishing operator, because the one that goes at sea every day, almost every day, are the fishery operator. So we cannot contrast fishery operator, we need to work with them. And they are the ones that know the sea better because they are at sea. And so asking them, when you fish, can you please collect 10 liters of water and filter it, but in sterile condition, please. Do it in Chioggia. You cannot even finish the first sentence. But if you provide them something which doesn't cost, which doesn't impede their activity, maybe, sometimes, not always, they will do it. So this is just a ball, a printed 3D ball. You put batuffolo, a gauze inside, which was sterile once, and you put it in the net. After the fishing activity, the fisherman take the ball, put it in a pocket, in a plastic bag, and no, sorry, not in a plastic bag, in a tube with alcohol, ethanol, or silica beads. So alcohol, you don't have problem to transfer alcohol, even if it's not that big problem. And then from that you can extract DNA. They did several proof of concept study, and it's working. And we are using it. Many institutes which are doing scientific survey, like the MEDITS in the past, hopefully again this year, or other scientific survey. For example, in this picture, we apply this metaprobe. We used to call it the palle di Tommaso Russo, but it's not really sound very well, so it's called metaprobe. And we apply to deep water long line, palangari di profondità. Still, it has to be optimized, it's not working super good, but it's working. So there is some potential.
Magari the question on the
Metaprobe.
on the metaprobe. But if you deploy this metabole within a trawlnet, it ends up in the middle of a bunch of fish. And how can this be comparable to apply to a ferry, where so a ferry sample just water where the fish can be and pass it, compared to?
But Metaprobe was developed for fishery operator.
Okay.
So in a ferry there will be no improvement in putting the Metaprobe, no, because in the ferry the water is l'acqua che viene presa dentro is continuously, so you just filter that.
Mi viene difficile formulare la domanda.
No, sono io che forse non l'ho capita.
No. So my gut is in setting this framework, then you need to rely on fishers.
yes.
So,
Yes. But you need to collaborate with fish.
no, I mean this would be difficult to apply to a survey without a trolling net.
No, we are trying to see, so the MetaProbe system was conceived for demersal trolling, for medics. So now they are testing if putting it, normally it goes inside the net. Now they are seeing if it's better to attach it, come si chiama? La bocca, dove stanno i divergenti. In another position of the net, because so that it doesn't get into contact with sediment. Because the result of the MetaProbe study, they are published. The last paper from Giulia Maiello was published last week. Is that with this system you can get a good representation of the fish that you have in the troll, but also you can add other species, the pelagic one, but also some which are in the sediment. So you broaden the capture of the assemblages that you can identify. Not only the one that are caught by the net, but also more other species. And so maybe you can use it to monitor biodiversity or to see if there are species which are not reported in the landing. Now all the studies are done double blind. So you analyze the DNA and then you check with the catch data. Okay.
But this method can be really useful to detect the rare one or the one that, yeah, since they are very low in number, for example, the squatina. I remember from the presentation that they found quite rare species around the Swift area.
But also, if you know exactly which species you are looking for example, endangered, then you can analyze environmental DNA, but not by metabarcoding. You developed, for example, real-time PCR assay that are highly sensitive. So even if you have a few copies of DNA of squatina, they will tell you. So they are doing this also for great white shark.
Is it quantitative?
I would say no.
Okay. And how far it is from you?
How good are you in developing science? So we rely on you.
I think that the last ICES,
There was a session on it, chaired by Tommaso Russo and
Tommaso Russo say that they are quite close to
quantitative.
maybe
But how quantitative is truly quantitative at all? Okay, you can count the fish in the net, but it's really is relative to the catchability of the species, no? So it's not something, it's not perfect, you cannot try.
The combination of the methods probably
Can provide, yes. There are papers like in Mesocosm, they're really trying to create like a regression curve to estimate. So again, there is a huge field of research in this topic, and the goal is to have it semi-quantitative. But it's not even truly quantitative, for example, microbiota profiling, no?
No.
They are doing since 15 years. So they do it semi-quantitative. They compare the amount of, you can tell it, please.
Is it in percentage?
In percentage and compare to mock community.
Yes, maybe we can compare different environmental microbiome, but in percentage of
So there is potential. And also there is potential for engineering also this method, no? why a sphere? Because it's the easiest to think, but maybe we can think of other structure, or so there is still a lot to have fun with. We applied to shark. Our pet species are elasmobranch in the lab. We all love to work with sharks, even if we never get funding for sharks, so we must work on other species to have fun to do research on shark, and we apply it. But why we could do it? Because we have the reference database of almost all Mediterranean sharks. So we have tissue for a lot of species. We could develop the standard, so all the sequence, because environmental DNA for fish use another marker, a different marker. Okay, this is very technical, but we could develop the standard for 80 out of 88 elasmobranch species in the Mediterranean. So we have most of them. The 8 that we don't have are very rare, and so we don't know where to find the tissue. And we could then compare the catches with the information from the DNA. And you can see that DNA could detect, for example, I cannot tell it in there. You can read it. So more species in the DNA than the one in the catches. If you do it at species level, okay, but again, a short fragment, because with environmental DNA, you have very degraded DNA. You can sequence maybe 100 base pair. In this 100 base pair, you don't have enough information to resolve a very closely related species. So species within the same genus. So morphologically, you can distinguish Raya asterias by Raya clavata, but with a short sequence, you cannot. So that's why you see the difference. If you only consider genus, then at genus level there is complete overlap. And biological DNA could identify Pleuroplatypus violacea, which was not in the catch, but is more pelagic. So maybe it was nearby, but not in the net. And then we could detect Carcharinus leucas. So maybe is another Carcharinus species. Carcharinus leucas is quite an aggressive shark. So don't tell people from the Lazio coast that they have Carcharinus leucas in their water. But still, this is a species that was not never reported before in the Mediterranean. We take it. We argument that there might be many reasons why we detect these species, or maybe there is not enough resolution. But even if it's not leucas, the other species nearby are again tropical species. So is the same, is very unlikely as for leucas. But you know, elasmobranch are very migratory, so maybe he entered, and so that's all. But the very important study is that we can use this data to use elasmobranch by catch, elasmobranch diversity. It correlates very well with environmental information, fishing pressure, so we can use it as a proxy to infer, to characterize the community. Okay? Are you okay? Can we finish this part, and we have a break, and we have the wrap up? Okay. So we are already halfway, 6 out of 11, but the last one are very fast. So this is another, so species identification, which is highly correlated with the consistent monitoring. Now, so you cannot characterize the community if you cannot identify the species. Population structure, so identify the population and the associated stock, to manage properly the stock, to do mixed stock assessment, And now we need also which other information. The abundance. We would like to know how many fish are there. So how many fish we can take from there, or how many animals. So, and now there is a method which is estimating not, let's say, have you ever heard of effective population size? No. Someone has heard but doesn't remember what is it, maybe, I think. Effective population size is an estimate of the is a genetic estimate of the size of the population. So it's the size of the population that has the same drift that you measure in your actual population. So it's not the census size. It's a genetic parameter. It's correlated to census, so the number of animals, but it's not. And the relationship is in order of magnitude. For fish, it goes, there are from 3 to 5 order of magnitude between effective population size and real census size. But again, Cairo, research from Cairo developed a method which is called close-kin mark recapture or genetic tagging, which they say, and they proved, can effectively estimate census, so real abundance, from a genetic estimate of the number of individuals. So this is extremely important. And again, in Europe, they want to apply to tuna, because it, again, is the most important resource. And ICAT launched multiple calls for this close-kin tagging, but they all went deserted. Because it's very, to apply this method, they developed for southern bluefin tuna, Tunnus mccoy, and to apply to any other species, you need to know very well the stock structure. The phrase is highlighted in yellow, and you need to be able to collect juvenile from known spawning population. At the moment, it's not possible for tuna because we really don't know the stock structure, and we don't know the known spawning population. This method is very complicated from a computational point of view, but very easy to understand. So you sample at the same time parents and progeny, okay, adult and young one. You fingerprint, you genotype them at the genome level. You need a lot of marker because you need a lot of statistical power to do that. And you need to genotype a lot of fish, because then what you do, you reconstruct the pop, parent offspring pairs. So the more family you reconstruct, the smaller is the population. The less, the fewer you found, the bigger is the population. I'm going to make an example which is a bit politically incorrect. If you go to a valley mountain, if you go to Val Gardena, everyone's surname is Comploy, Senoner, Moroder. They are all relative because they are few and they create family among them. Now the population is small, so it's easy not to find someone which is a cousin or of someone else. If you go to Caracas, maybe you can stay in Caracas 6 months and you never found someone which is relative of someone else because the population is bigger. So this is exactly the same. So the number of family that you can reconstruct is conversely proportional to the number of individuals. It's just a probability. If you have a huge reproducing population, a huge spawning biomass, then it's less likely that you found the progeny of this, especially in batch spawner, let's say. In animal, if you have site fidelity, in other animal, you cannot apply. Was it quite clear? Okay. So they applied, for example, in deep sea fish, anglerfish, Lophius, they applied to it. They apply, you see the paper on southern bluefin tuna. They modify the methods, the computational methods, in order that you can use only the progeny. If you cannot sample the parents, they modify the method, then you just assess the sibship. How many fish are brother or sister or cousin or so on. So and again, the more they are related, the smaller is the biological, the number of individuals which are effectively reproducing. So they apply to white shark because it's not so easy to sample adults of white shark. And then there is again a very important review which I describe all this method. They are called close kin. So mark recapture is a common method. Now you mark and then you recapture. Close kin means the one that are close relative, I parenti vicini. Now, again, ICAT is still attempting in doing this for bluefin tuna because they did it in the Western. So the American did it and it's quite promising. And so they are now thinking of doing it, applying only to larvae. Even if it's quite biased because normally larvae are from the same individual. So it's high probable that larvae are related, that you find sibship among larvae. They did it in the US and they say they found a low level of sibship. So if you're interested, next week, you don't have a teaching week next week. Do you want to go and relax? No, you can participate to the online meeting. There is a workshop next week. It's online, everyone can attend or go to Madrid on bluefin tuna close kin mark recapture. Or to register, I think. Because again, this is one of the other topics in which EU funds are drained. I mean, really. So we want to monitor in time. Someone was saying monitoring a long time, no? Someone was speaking about fish industry revolution. We are getting there. So because, sorry, I forgot to say, we are now in the impacts of fishing. So from environmental DNA, we changed the big topic, now is impact of fishing. So the number of individuals on which fishing is insisting, and also estimate the implication of this fishing pressure. So monitor through time the genetic diversity. If a population is overexploited, its genetic diversity is depleted. And so, again, you can assess it by estimating this genetic value of the size of the population through time, N is called. And you can estimate it the contemporary, the long term. It all started here, in this building. I don't know if, well, not where you were seated, but in the room here, because it was possible, because in this building there was the collection of Massimo Sella. Massimo Sella was, is that now, but he's from the same family of the Banca Sella. Have you ever heard of Banca Sella? It's a bank in Italy. It was a very wealthy family. They owned a lot of tuna traps around the Mediterranean. They were originally from Genova, and Massimo Sella from the Sella family, he was also a naturalist. So it was the time of Ghigi, and he was also running a naturalist museum in Rovigno. when it was still Italy. And then, because of the war, he had to escape. And all his, come si dice beni personali in inglese? Property. Okay, but also the money, all the were being taken because of the war. So he packed everything from the museum and he shipped it to his colleague Scaccini. You know who is Scaccini? There is a picture of Scaccini also. He is the one who this also building is dedicated. The boat, Andrea, is named after. So he was the former director of the marine biology, and so there were boxes full of tuna bones here. Oh, and one day, oh, what are these boxes? Opening, oh, tuna bones, and that's why we started. So my colleague started. I was not here at the time. I was partying in Marina di Ravenna, and they started to analyze the DNA, extract DNA from bones. So do this estimate, go back in time. So genetic, you can have it in space, so geographical level, but you can have it in time also, backward in time. It's not only us doing it from bones. We never discover, let's say, nothing new. It was already something that was applied, for example, to other species for which you have longer time series. Time series of what? Not of tissue. It was not the common habits to put tissue in ethanol in fridge, but of what? Scales and otolith for age reading. And you can extract DNA from the molecular tissue, from the tissue remains which are around scales and otolith. So they did it in cod. They started doing it in cod, but also in sole, in flounder, in many species for which you have a collection. And normally the body part that you have the biggest collection over time are the one that you use for age reading. So le scaglie, gli otoliti. Everyone knows what is otolith. Otolith is a bone structure. Help me. No, it's not bone. Sorry, it's a hard structure. Sono come dei sassolini, come quelli che abbiamo noi un po' per il suono nelle orecchie. Sono come dei sassolini nelle orecchie dei pesci. Calcified structure. And they grow each year, and so they create rings like the shell of the clams, and you can use it to age. That's why they were collected since ever they were collected to age fish. And now you have this huge collection, which are like on paper. They were just sticking them on paper and keep it on the shelf. So it's still possible in some cases to extract DNA. The DNA is not good quality, is not good quantity, is very fragmented, but still sometimes you can infer information from this study. Or you have, in other cases, you have bone or spines or other remain. But still, some species are particularly challenging perché moving, from microsatellite, also here on SOL, to genome-wide data. So you can do genome from ancient DNA. Again, we learn it from Svante Pabo, no? He sequenced Neanderthal, Denisova, and we can still sequence tuna from very old time, not fossil. One of my students said, no, fossil is not organic. You cannot sequence DNA from fossil. And despite century level of catches, we don't see genetic erosion in tuna. So we confirm the result obtained with microsatellite. Maybe it's us. We are not good in doing this. But again, for cod, and again, cod went through a dramatic decline Still, we can. It seems that there is genomic stability through time, despite everything that happened to cod. Now, cod was collapsed locally and extinct, and so on. So this is promising, but not for all fish. So they did it in other fish, for example, fish with a more limited geographical range, and then they could really see a genetic erosion. In terrestrial species, you see it, like all the endangered species. You can even quantify the genetic load due to this deleterious effect due to the low level of genetic diversity. But still, in fishery, this is still difficult. But very interesting if you have access to long time series. If you have access to long time series and phenotypic data, you can also try to test, or if you have a scientific question related to what is called FINE, fishery-induced evolution, FINE, which is the evolutionary response to fishing. Why is evolutionary? Because it's changed at the genomic level and then is transmitted through generation. Why so? Because there are evidence that over time, the phenotypic trait, the life history trait of some highly exploited fish are changing. Normally, they grew less, they reproduce early, and they reproduce when they are smaller, lowering their reproductive output. Many fish, they just are batch spawner, they put gametes in the water. But the number of female gametes that are produced, they really do depend on the size of the female fish. So again, there was a big European project called Fishery-Induced Evolution, because again, for cod, they could really, having a long data series, they could estimate that cod were becoming smaller, smaller, and smaller. The average, the distribution of size classes was decreasing. So normally in the 30s, a cod of 9 years was 85 centimeters. In the 20s, a cod of 7 years was 63 centimeters. So really a big phenotypic change. And also with the eggs, the quantity and quality of eggs was decreasing. So there are, now I enter a field which is not genetic. This is ecology, fishery science, the Bomber-Tallaffi equation. Okay, and so model say that the output, the possible explanation given by this model is that if there is a size selective fishery, normally the fishery is size selective, the bigger, the bigger one have a lower survival, they have a higher catchability. And so if you reproduce very late, if you wait to be obviously a fish doesn't think, so it's not deterministic, but reproducing at a high age and big size is not always the winning situation, because your probability to reach this age and size is very low, because you will be fished before. So reproduce as soon as you can, at least you reproduce once. And so they are growing faster, and as soon as they reach an age and a size in which they can reproduce, they start reproducing. But the point is that if you start reproducing, your energy allocation goes into reproduction, and you don't grow that much anymore. So the fish remain smaller. The L50, so the size at which, okay, you know what is L50. So the standard size at which they reproduce is smaller. It means you produce less gamete. So your reproductive output is lower. So it's really a tragedy. But so this is really affecting, and this is what they say. They saw it in many flatfish, in cod, in whiting. You have seen it. Your colleagues saw a very clear shift in size. And so now the important is to understand it from an evolutionary point of view. So again, sorry, it all started in this building. These were the boxes. These were. Sì, certo.
From this approach, can we also see if there is some phenomenon connected to epigenetics instead of genetic variation?
For sure, there will be something connected to epigenetic, or also maybe not only modification in the coding region, but most likely is a modification of the expression level. But still is under studying. So if you want, I have more example, and there are a lot of study on, again, code, but also a recent study, a manipulative study on a small, is a fish from that is in America. I can provide it to you, where they see that they saw the same response, but different genetic region affected by it. So it's still far to be very clear cut. So it's not so non sono i piselli di Mendel che aveva preso quel allele, no, quel tratto quantitativo. So again, sorry, it all started here, you understand it, but I really wanted to show that it's this building. Can you see it? Behind the big Greg and behind the big boxes. You have met Greg? No, never? Okay. Just Adam. It's even bigger than Adam. So these are 2 or 3 boxes. But then they also had a lot of fun, because it all started from the Massimo Sella archive, but then we really entered into Indiana Jones, archaeogenetic. Thanks to the collaboration with the Ancient DNA Lab. I think Professor Luiselli mentioned it to you this morning. In Ravenna, there is an archaeogenetic group. And so this lucky guy went sampling in the Cadiz area, Tarifa area, because there were all the tuna tracks from the Roman, from the Iberic. They also went to Constantinople because, to Istanbul, because when they started the work for a new metro station, they found the remains of an ancient Constantinople harbor. And in the harbor, there was the slaughtering of the tuna. Now they were processing the tuna and putting all the bone aside. So they are digging in the waste bin, in the ancient waste bin, where they can find all these big stones. And imagine that in some of these Roman settling, they were using tuna vertebra. Tuna used to become giant tuna. They were using tuna vertebra as building material. I muretti a secco con le vertebre di tuna. And so these are our colleagues sampling in archaeological excavation, and then drilling the bone in a more appropriate place to extract from the inner part of the bone powder, and then process it in the ancient DNA lab. So this was possible because of several funding, because it cost a lot. It cost a lot, so we were my boss, the professor, the PI of the team, Professor Fausto Tinti, was involved in many projects that could finance this archaeogenetic work. And recently, Adam Andrews, here is this contact if you want to know more from him, is a PhD student from a Marie Curie network. So thanks to this Marie Curie fellowship, Adam could really perform a giant leap in this research field. So because he could, these are some of his papers, And it could have a very multidisciplinary integrated approach, assessing both morphology, so he reconstructs size from bone, from annual ring. It could analyze a stable isotope, so a more ecological and trophic approach, and also whole genome sequencing of ancient and modern tuna. So it was brilliant to manage to extract good DNA for sequencing from samples that are dated 300 years before Christ. So for the moment, results from the morphological study are all published. Stable isotope will be published soon. So, and indeed, over these 2 millennia, they are growing faster. Nowadays, they are growing faster than what they did before. And age 1 and 2 grow faster, even faster, with respect to the 1600, and they coincide with the stepwise increase in surface temperature. So it's really correlated to ecological, but also fishery, because tuna has been fished not only now, but has been fished also in the past at very high rates. So in the past there was a period between in 16 and 1800 in which there was an explosion of tuna trap, and we have records of it because the church was putting taxes on the tuna trap. So we have more or less information. So the PhD of Adam is really challenging because it's really archaeological and also using source of information which are not Excel table or data frame. Getting information, when you go back in time, you need to get the information that were recorded, and normally is when you have to pay taxes. They were recording each tuna that was caught, because then they had to pay something for this.
Yeah, but it's incredible to can deal with this kind of series. How we are used to work with a few years when it's very hard to find a meaning, because you are not, here almost a snapshot. So it's great to think to limit to up to 2,000 years.
Yeah, but you need quantitative data. These are more qualitative. So it's more like local ecological knowledge. So
even qualitative can be.
Can be.
If you are biased in your quantity, also qualitative is
No, but they did a lot of work, and obviously you have it for tuna because it's moving so many. I don't think you have record for Tracurus. No. Quale?
No,
Questa? Questa? So I'm not an expert, and I just followed the genomic part of his PhD. But if you are interested, there will be the final conference of this sea change network. It is really not only tuna, but there are people working on small pelagic, but I think ring. No, but also mammal, turtle, all marine species affected by changes through time. So it's really nice because it's science, but it's also science from genetic, isotope, but it's also from archaeological science, from morphology, and so there will be the final conference in Ravenna in 2 weeks. So, I think it will be also online, because not everyone can be in person I don't have the Zoom link now, but there will be for sure a online link, and just remind me, send me an email or ask the coordinator, I can share the link to attend the main talk. Billfish, so it's not only tuna, and also ricchechi, come si dice? Walrus. There was a PhD student analyzing the tusk. Okay, I can feel you are tired. I feel it in the air. So, okay, this is more about aquaculture. Who is involved in aquaculture here? Qualcuno c'è? Which species? Okay. How often are accidentally escapees reported in sea bass and sea bream fattening cage at sea? Ci sono ogni tanto dei report in cui dicono che ci sono stati dei rilasci inintenzionali, che i pesci sono scappati dalle gabbie a mare. È un problema perché can you please explain to your colleague why this is a problem? When they escape, most likely the farmer release unhealthy animal. When there is an outburst of some infection and the density is too high, they release animal, but they say it is unintentional escape.
It's probably related to the gene of the species, optimized species.
Exactly. Because the farmed one are strain which are selected, no? And if you release it to the environment, what happens? If they survive, they will reproduce with the wild one. So it's very important to avoid the unintentional releases and to be able to track them. If you can track them, you can find them. And maybe next time they are not doing it anymore. So there were at least 2 projects. No? Ho detto qualcosa che non off topic dopo. So, but aquaculture, so we cannot, we are not hunting for deer anymore. We have farmed animal on land, and so we need farmed animal at sea if we want to feed the human population. No? So aquaculture is not bad. It's bad if you have bad habits, like, but it's less. It's producing less CO2 than than land farm. So there are a lot of benefit in aquaculture, but sometimes you have to monitor these SKP. The tools are mostly the same. They do genetic selection on the strain, so they have fish that are selected for reproducing and producing larvae. They have a nursery where they grow larvae to juvenile, the fries, and then they sell the fries to the aquaculture. Intensive, extensive, inland, at sea, in ponds. So there are many different points. So what can genetic do in this? Can trace escapee. For example, for salmon, there is a database, a data bank of genetic fingerprinting of all main aquaculture facilities in Norway. So when there are escapee, they can simply say, okay, which is the farm from where this fish come from. Okay, we go there and we assess it was because of a storm or maybe predator that are trying to eat the fish inside the cages, or it was a bad, not proper management of the farm by the farmer. And so genetic can help in this, can trace it, and also if you can demonstrate that you are following stricter rule, then it's also good because you can also enable your farming with some quality level, which are not compulsory, but for the consumer, they can be an added value, like responsible fishery, no? MSC or Friend of the Sea or many other. There are now a lot of them, Adriatic responsible fishery management, but there is also aquaculture responsible practice. So this is very important because you can also monitor yourself, and it's like organic food, no? It's an extra cost for the producer to have a return from the consumer, more or less. Okay, pathogen and invasive species. I think you had a full afternoon now of microbiome, the lecture given by Gianmarco Luna. I think it was Tuesday. I could not follow any, and it's a pity, but I just realized they are all recorded, so I can listen to them. So we are getting to the end. I know you are happy. And what happened post-harvest? Again, a fish doesn't carry an ID. A fish cannot tell you where does it come from. But the regulation asks you to provide all the information. If you go to the supermarket, big retailers, they are doing it because they know they must do it, so they have control and they do it. I don't know if it's correct most of the time, but they are showing it. They must show common name, so commercial name, scientific name, way of harvesting. So if it's pescato allevato, so harvested or farmed, fresh or towed or someone else, but also the fishing area. So where does it come from? You have to rely on the chain of trust, no? From the fisher to the gross retailer and because there's no way they are trying to use like blockchain or other system, but still you can, it's only documental, no? But with DNA, you can, in some cases, for example, for sure, verify the species. So you are not going to selling me Pangasius for another higher value species. No, for sure you can, with the molecule, you can clearly detect substitution fraud. It's more challenging to detect fraud of origin. But why they should create fraud of origin? Because sometimes they are going to fish in protected area or fishery restricted area, FRA, or they are fishing when or where there is closure to fishery. So, come si fa a avere tutto quel pesce fresco in Adriatico? I'm telling to the one from in agosto. Hai voglia che sia della piccola pesca, no? Perché la piccola pesca può pescare, no? Ci sono delle eccezioni. I cannot, so if you go to all the fresh food, freshly caught during the summer fishing closure, is very unlikely. There are some fishing here that can fish during the fishing closure, but the summer is also the period in which we have the most tourists here in this on the Adriatic coast. So the fish is not enough to feed all the restaurants that expose fresh sole from today and so on. Or if you are exceeding your quota, now you can. So there might be several reasons why they misreport the origin of the fish, and with DNA we can track it, because if the species has structured population based on geographical scale, based on the geographical range. So if it's a highly migratory species, it's very difficult, no? Tuna is very difficult to tell if it's from Atlantic, sorry, fish from the Mediterranean or the Gulf of Mexico. But other species, for example, sole is very boring. They cannot do much, it's not highly mobile. So you can identify sole from the North Adriatic to the South Adriatic or but not only sole. This is an example of 4 species, and this is the number of SNP which are required to have 99% accuracy in assignment. So forensic level, you see, is very low. So you can develop a tool which is fast, it doesn't cost that much, and you can really have a solution. And there is a case, not in the Mediterranean, but in Denmark, in which indeed a fisherman, it was not brought to jail because it was just given a fine, but the fine was given after a court process, and in which the proof of evidence were from a genetic assignment in Denmark. Maybe in 50 years also here. Or maybe we are so good that we don't do any fraud in 50 years. Okay, so you see, when I was telling you at the beginning, we have the tool, but the end user understands the solution, and the tool is really easy to apply and to have an informative answer in a reasonable time. You cannot say, I have this quantity of muscle, and I want to check if which is the coli load, or if there is some pathogen. Yes, give me 2 weeks. I extract DNA, I send it to sequencing, I wait my Illumina run, and then I process my terabyte of data. No, the muscle are rotten in the meantime. So you need something which is also fast. But you can derive this fast and accurate tool only if you have the scientific background. And this is the paper. So coming to the end, I propose you these 2 papers, which are really putting genetic into applied context. So genetic under the common fishery policy. I know this is very thematic for those that works on fishery or resource management, or can estimate of genetic effective population size contribute to stock assessment. So can we produce data that can really enter into the process of stock assessment? And last but not, a lot of, as I say, a lot of paper, review, workshop, really on this topic, genetic and management in marine, so genetic and fishery management, but also, last but not least, is it cost-wise? So this is a paper from the JRC, the Joint Research Center, really assessing if we can really go toward, is worth going in this direction, or if it will remain too costly. Because if we cannot implement it, so like ecolabel, they are useful, but can every fishery afford having an ecolabel certification? At the moment, no, it's too costly. And the reward from the consumer is not enough to pay for it, at least.
Yeah, this is the problem now with fish products rather than products from agriculture, to have an increased price compared to the conventional.
Okay, but this paper say yes. No, sorry, this paper say it's not too costly. There is hope we can continue. As I promised, I finished. So do you want to have a break and have a kind of wrap up after the break? Do you want to have the break and go home? Do you want to have the break, the wrap up, and then it's done? Ai voti, c'è la rappresentante.
Maybe a short break, 10 minutes, and then we go back.
You decide.
I would prefer to wrap up and
L'abbiamo messo ai voti, very democratically. The wrap up is super short, super short. It's just that to be sure that you are all well awake when you exit, you don't get the wrong train. You have to come giù Cascasella, no? Per svegliarvi. Dopo gli spiegate chi è giù Cascasella. So we did it. Okay, we finished because now I gave you example on how genomic tool can now tackling several challenges to ensure sustainability and profitability of fishery management. We did it. I'm surprised too. So, which is the ever most powerful device created by human? I hope you didn't see my slide before. L'hai visto? La panda all-wheel 4x4. How do you say 4x4 in English? Four-wheel all-wheel drive. I don't know. Do we all agree that this is the most powerful device ever?
Maybe not yours.
Yeah, but I needed the panda because 4x4 is the solution to every problem. So 4x4, 4 basi, il DNA is 4 bases, and it can solve 4 problems for fishery management. It can tell you species community, it can tell you population stock, maybe in the future it can be very good to tell you age and abundance. No sex, not yet, okay? No study, not maturation stage, but not always this information are retrieved from, for example, for landing or from fishery statistics. No, these are the main that you need for doing stock assessment.
Yes.
Federico, no? For your stock assessment, what do you need more?
After the panda,
You're still thinking on the panda. So, but from my small knowledge, from my limited knowledge, these are the 4 main input that you need in stock assessment, no? Age classes. Okay.
At least the maturity you can observe with a certain degree of certainty by eye, while abundance and age are something that are really less reliable. But you can specify the age reaching, and it's very difficult to have a proper estimate of amount.
So I hope that in this long afternoon, I convince you and I showed you the promise of the 4 bases of DNA in answering trying at least to answer these 4 needs in resource management. So species community, you can do it with barcoding, beta barcoding, from gut, environmental DNA, from whatever you want. Defining population and stock, we can do it with different population genetic approach in space and time. Age, epigenetic is very promising, and abundance, again, there are methods that are based on population genetic, but are based on genetic profiling, like the closed skin marker capture. We didn't explore adaptation, that you can do it with transcriptomics, response to environment, because these are more ecological or physiological. Now, today the focus was resource management. That's why. Martina was quick. Now, at the beginning, I asked you what can genetic genomics do for aquaculture and fishery. Now I would like to ask you, what can genetic genomics do more for your PhD project? I hope you could get some hints or maybe some idea, or maybe now you are 100% sure that you don't want to have anything to do with genetics. Eh? So I don't know. It was open for the last question. No answer?
For my PhD, it would be very useful because we have this issue with age, so if you can develop better kind of in that direction, it would be great because we already are a mess sometimes. And also to reconstruct the ancient sites of the stock in order to understand how the growth curve has changed with the time.
So you need to look for a good molecular biologist in your team. In which institute are you?
CNN.
Ah, you have plenty there. Other?
For me, that I work in microbiome can be a good option to couple with the microbiome genetic and the fish genetic. So maybe for maybe having this kind of answer in the fish variability, we can couple with the microbiome, so it can be an option.
And on which fish are you working, if I can ask you?
I work in Seoul and Sardini.
In Seoul? Seoul aquaculture?
No, Seoul from wild Seoul.
Because you know my secret desire is to establish Seoul aquaculture in the Mediterranean.
I saw many papers about that, but no one is able to do a complete cycle, I guess. I don't know, for common soil. For
common soil, yes. We need to I tell you afterward. We can have our business. With who are you doing your PhD? Sorry. Which is your supervisor? Ah, okay. And, there is one species of sole, which is called Solea gypsiaca, which is totally identical to common sole, and there were successful results in the Orbetello Lagoon. There was one farmer that could raise Solea gypsiaca. He didn't know it was Solea gypsiaca. He thought it was selling and raising Solea, but they are cryptic. It's impossible to tell them apart. There are only 3 people in the world that can do it, and they are at the CNR, Laura Sabatini. No? And Laura Sabatini. Giuseppe once. Giuseppe, sorry, Professor Scarcella, no, he can do it. Igor Celic, maybe.
It's a long time that Igor is not coming with us.
So they are completely identical from the external morphology. They taste the same. So maybe in the future. Okay, I think we can close by 6. Now the last day. Yeah.
So it's 5 to 6.
So 5 minutes more of question or curiosity or feedback from this.
It's useful for my PhD as long as I can quantify how many animals of a certain species I have. And I know this is a hot topic.
Relative quantification. Which animal?
No, animals of number.
But there is the possibility because you have a standardized volume of water, no?
Yeah.
Then I think so, because in mesocosm they are doing it. And you know also which species you want to quantify, or you want to quantify it, so you want the diversity and the quantity associated to each species.
I want to avoid sorting.
Avoid sorting, then metabarcoding. But then if you really want, then maybe you can say there are the more crustacean or more, it depends on the accuracy you need on It depends on the accuracy you can achieve on your sorting. Can you go species level to the sorting?
It depends on what kind of looking at. Some profiles are easier than others.
But in the framework of the PNNR, come si dice in inglese?
National Recovery Plan.
In the
And the resilience.
in the framework of the nature future. I'm very tired.
Future Biodiversity Center. National Biodiversity Future Center.
There are
National Future Biodiversity Center.
Whatever.
Okay, future is associated not to the center, but to the biodiversity.
Okay. This one. There are two spokes related to marine science in which I think many of your supervisors are involved, and they are establishing omics observatory in which they are really developing this kind of tool. And for sure, for plankton is one of the mainstream topic because plankton survey of plankton are very well established. I think that there is a paper, maybe you have seen it, in which they compare the result from the continuous plankton recording, you know, now and metabarcoding result. So maybe there is
the
identification, but also relative abundance. So I think you are in a good. Which is your supervisor? CNR? No. So you can for sure interact, or I can give you some name of people. For example, the colleague at the Antundorf station, Sarno, Diana Sarno. She's doing this zooplankton analysis, but also I know is involved in this effort. Also, Valentina Tirelli from OGS. They are comparing plankton survey with metabarcoding result because the avoid sorting is something that.
Because it's long and physically and mentally exhausting.
I did it few times, but I perfectly remember things. And I was sorting invertebrates, so bigger animals.
Okay.
Okay, so
Alessia, I guess you win the challenge.
Che challenge?
Of keeping people awake Friday after coffee break.
If there are questions from online, tell me if you can still hear me. Ciao. Hello.
At least in the room they are awake. So,
yeah, they are laughing. I don't know if of me or me or.
I was sure I did the right choice.
Chiudo.
No, leave it to Mariana. So apparently, actually not apparently, we are done with this 2023 teaching week. So now you are free to go back home or to your lab, whatever you want to go. And it was a pleasure for us. I hope I heard some noise that you also had fun a few times during the last couple of nights. Someone told me. Someone told me, you know, and that fun was part of the program. Actually. So stay well, good luck. I wish all the best from my side and the academic board member. We will meet again here in presence, face to face, from September to December, depends on the different cohort of students for the admission to the next year or to the final exam for students that are at the third year. So good luck. I'll see you soon. Bye bye.